Welcome, everyone. This is a very special day. This is the Department of Neurological Surgery's Research Day, and it's fantastic because so many people working together come together and learn and basically embrace what we're doing, which makes a great deal of sense. I recently wrote a book, and there's a chapter in it called "Why Do We Do Research?" It allowed me to reflect on a lot of things that really bring us together as a team because we're as diverse can be, big and little, all races, types, etc., coming together for common themes and coming from very different angles to it. And so if you're a clinician and doing busy surgery, many people succumb to saying that's good enough. If you're a scientist and you're just doing basic science for the love of science, that's very good, but not quite completing the circle. The Latin word for integrity talks about the wholeness of something and including everything. And it began to make sense as I thought about this, about how important it was for the clinicians and scientists to come together, because that's what our patients need. We deal with some of the most phenomenally important disorders of mankind that relate to the brain, spinal cord, and nerve system. And if you think about it, those diseases, including cancer, including stroke, including trauma, including congenital anomalies, including pain, affect every single family, and actually most of us during our lifetime in the world. It's a worldwide issue. And so when we begin to attack this problem, we do it only by doing it together. And as I reflected when I was writing, I realized that my clinical work was inadequate. It is the best in the world. Believe me, that's what we're teaching. But it is inadequate for people with brain cancer, with stroke, etc. We have to do it better, and that's called research. And you do research by collaborating. And when you do that, you actualize those researchers who for all these years have been working on a molecule, and suddenly it becomes pertinent to mankind, to health. The integrity of what we do happens. And if it's any good at all, we teach it. And suddenly that is the integrity of what we do. So this is a wonderful day. We come together, look, talk to each other and say, I didn't know I was working on stroke or a brain tumor. I thought I was doing epigenetics, and yet we can make sure that that makes sense. That's what we're doing. It's a wonderful, wonderful opportunity. Probably, certainly one of the best days of the year for this department. So you do it. Thank you and welcome. The format, of course, is lots and lots and lots and lots of presentations in different formats. If you look carefully, you'll see that's everything from undergraduate students to really crusty old senile professors, okay? And that's great because they all need to get that perspective and excitement. When I was a student, having a professor just say something nice about my research like made my whole life. I remember it to this day, and that's very inspiring. Well, guess what? The professors are just as scared and they want to do it just as well, and you reaffirm them. So congratulations on that. And you think about research, it is not some segment of your education. It is your life. It takes place in the OR, in the lab, in the clinic, on the Native American reservation. Why not? Why not do it better there while you're serving? And the scientists come with. It becomes very important. So that's really why our department looks like this. That's really why the clinicians have to walk through or around the lab to get to the OR. That's why we bump into each other. That's why we forced the university to bring all the scientists to the WIMR, all the bioscientists. So imaging's there, so basic science is there. Things that we didn't even realize were pertinent to the clinical course. That's what we're going to hear about today. And we'll start with our really flagship for the year is the Gilboe Lecture. And that is a basic scientist, either from here or elsewhere, comes to really start off this. Then we'll have a series of lectures by people from the neurosciences here. And then probably the highlight is the fact that we combine presentations by poster presentations by everyone with food. I think that's brilliant, if you ask me, because it makes the most sense. But in keeping with the format of the last 16 years, this particular lecture that starts our neuroscience presentation, our research day presentation, is named in the honor of Dr. David Gilboe. And Dr. Gilboe really founded basic research here. And his career was long and prestigious. Like many of us, he came from simple background, military background as well. But like many of us, he was an outstanding teacher. And his research lives on not through his great breakthroughs, but rather through the people that he trained and the education he brought up. And that's what we honor today. We have a great honor of having Dr. Ann Stowe come to talk to us today. Dr. Stowe is from Kansas, and we'll overlook that for the basketball season because it's really a very nice people. And very importantly, her undergraduate degree was in fine arts. And if you think, why does Dempsey always call research the creativity part of our academic life? You can see where that comes from, because that's what you are when you do science, is you're creating. She then did her PhD at Kansas with a postdoc at WashU. And after a brief sojourn at UT Dallas, Southwest, she's been on the staff at Kentucky, where she has risen this year, congratulations, to professor. She works for several centers there, the Stroke Center, the Sanders Brown Center, and she is an integral part of their Stroke Center, which has a unique history because prior to that time, stroke was really sort of a backwater of clinicians because it was a desperate and forgotten field. People had it, they were sent to rehab or to the grave. But people began to look at science, and a young assistant associate professor there formed their first stroke center. And pretty soon everybody had NIH grants, and this grew and grew and changed formats. And that was almost 40 years ago, and that was me. So in 1988, founded that. So that was the grandfather of the program that you're in now. And it's great. Her work is in adaptive immunity and stroke. That is to say, how does the brain try to heal itself and how can we harness that? And she's looking at the mechanics of that. That is what happens in your immune system when you have the antigens created by a stroke. Do they trigger a process? Can we harness that? Can it lead to recovery? You know that we feel that is the big growth area in neuroscience. We've been doing nothing but stopping disease, basically putting seatbelts on people. Now, why not repair the injured brain? This is very exciting stuff, and we're very, very happy to have her. And it's a delight. We were able to meet and talk about different back roads of Lexington last night at dinner. But today we're going to hear about science, and we can't thank you enough. Thank
you. Okay, can everybody hear me okay? Thank you, Dr. Dempsey. And thank you, Dr. Vimaganti, also for inviting me here today. I'm very excited. And I have always tried to bring clinical research into my science. And it is a challenge when you are a basic scientist to even get funding from NIH or other, unless you have a great team of clinicians with you. So I'm hoping that by the end of the day, I will have piqued your interest in looking at some biomarkers of the adaptive and innate immune systems, as well as potential collaborations. So with that, my disclosure information, these are my current funding sources. And I'm also co-founder, equity holder in Cerilux. Actually, This is based on some of the clinical research that I'll show you from Backtrack, and this was the brainchild of Dr. Justin Frazier, who's a neurosurgeon at Kentucky, as well as Dr. Keith Pennypacker. So we've been looking and patenting some aspects of biomarkers in our stroke population. And to begin with, this is not done in a silo, and definitely I am not the only one doing this. So I would like to first thank everybody in my lab who has been here. This is a picture from Tuesday in Washington, D.C. at SFN. So I had a lot of trainees there. I also have collaborators in the Center for Advanced Translational Stroke Science. Okay, so I will be talking throughout this about some of the challenges that we have in studying stroke-induced inflammation. And I think that this is critical, actually, for understanding long-term recovery from stroke. But it's a matter of really trying to understand the patient-specific aspect of this, because we can do this in mouse models all day long, but if these are not relevant mechanisms in a stroke patient, it doesn't really matter. So I'll go through some of the background to hopefully have everybody on the same page, and do a little bit of our mouse studies. And then I will go into clinical studies, particularly some data from ischemic stroke and subarachnoid hemorrhage. I thought I would pare this down and keep it more focused. So I am not showing, we also do work in the adaptive immune system in brains from our rapid postmortem brain donation program, but I'm not going to go through that today. And then a final little biomarkers and why this really matters. So, as you know, about 800,000 individuals have a stroke each year, and Kentucky, which is right here, is in what is known as the stroke belt. So this is the highest mortality rate in the country, predominantly, and a lot of this is because there are rural and underserved populations within this area. So I teach a lot of stroke pathology courses at the university, and we talk about how our populations are very different than large city populations, particularly on the coast. So with this, you've got a stroke death that occurs about once every 4 minutes. And of these 800,000 strokes, about a quarter of them are somebody who has had a second or a third or a fourth stroke. So this is really also something that drives my research, because our immune system will respond to the first stroke. And depending on how that goes long term, same mechanisms that would mediate a vaccine will also shape and mold how you respond to a second or a third or a fourth stroke. So I think it's very critical to understand what's going on. And for those in the audience who may or may not know, there's no real pharmacotherapy approved for post-stroke recovery. We have mechanical thrombectomy, and we have TPA and tenecteplase, so chemical reduction of an ischemic clot. There are some surgical procedures for hemorrhagic stroke as well, but nothing long-term. And this is going back to, it takes a very long time for your stroke patients to heal from stroke. And this paper actually came out the year I defended my thesis. So for the trainees in the department, or here in this auditorium, you know, I was so excited, I'm going to do stroke research, and then this came out, and it really hasn't gotten any better today. We still have just one pharmacotherapy intervention and then a surgical intervention. So this is where we're starting. And a lot of this is because we're looking at acute stroke time points, again, going back to what is happening long term, because your patients take a long time to recover. So here you have initial injury, you have your deficits. In this case, this is a motor function. Immediately after stroke, you lose motor function. And over the course of time, about up to 6 months for a stroke patient to have spontaneous recovery from the injury. And what drives that early on is plasticity. So changes in the surviving neuronal networks and other areas of the brain. And I'm not going into a lot of details on this part other than some of the mouse data, but plasticity is what, so you have rehabilitation options here, and you can actually then do rehabilitation longer term. But what I'm focused on is the neuroinflammatory aspects of this. When does it occur? Where does it occur in the brain? I'll show a little mouse data for that. How is it induced? And we talked about antigens and activating the adaptive immune system. We've studied that in the lab, but I'm not going to talk about that today. And why is it happening in one patient versus another? Each patient is going to have a very different inflammatory response to stroke. So it's not tailored and it's not easy. And this contributes to why we don't have really a good consensus on rehabilitation for stroke patients or on any pharmacologic adjuvants to rehabilitation to improve long-term recovery. So this is your background slide for anybody who has not had a lot of exposure to the immune side of things. The adaptive immune system ramps up in the first days after stroke. So you have your innate immune responses. These are not antigen specific. They are just a general response that occurs to injury. You cut yourself, you have a bacterial or a viral infection, and this also occurs after stroke. You have a big innate immune response, meaning that in stroke and any other type of neurological injury like TBI or spinal cord injury is sterile inflammation. That is what it is considered. So you have an immune response from the cells that are injured or dying, but it is not driven predominantly by bacterial or viral infections. What happens after the innate immune system gets upregulated is that in the course of days, you actually then have adaptive immune mechanisms. So these are by B lymphocytes, which we'll go in a little more detail about, that produce high affinity antibodies. So as well as CD8 T cells or cytotoxic lymphocytes. These are critical for killing tumorigenic cells, for instance. And then your helper T cells or your CD4 T cells that work in conjunction with B lymphocytes and also help mediate this. And just to orient you, throughout this talk, these cell types will actually be a little symbol in the lower corner, just so you can try to, so I'm not overwhelming, because I know sometimes you get, it's very complicated with the immune system. So what happens after stroke? You have loss of blood flow to an area of your brain, and you immediately have excitotoxicity, ROS generation, mitochondrial uncoupling, everything within the infarct and peri-infarct region, blood-brain barrier disruption. They can actually then activate astrocytes and microglia within the area, and this is one of the major cell types that are actually studied in neuroinflammation. So a lot of the innate immune cells within the brain and how they respond to injury, they can produce pro-inflammatory cytokines, which in and of themselves can induce neuronal death, but it also recruits initially neutrophils and macrophages, so our innate immune cells within the area, and this is a feed-forward mechanism, so this can increase the size of your infarct. If, however, you are young or you exercise and you don't have heart disease and you have a regulatory response, that can actually shrink the infarct. So if you think about even the initial injury of a stroke patient, that is not static. That changes and can be modulated by your immune system over the course of hours to days after stroke. What I, though, am looking at more is this conversion between the initial injury to your neurovascular remodeling, your plasticity, your new blood vessels that are growing into the areas, and the changes in the existing structure of your neurons that survive, and how our long-term immune cells, B cells, and then our CD4 and CD8 T cells, how they can actually influence this. So I have this set up as having some challenges involved in this also, because I think that the more we highlight the challenges and hopefully the more we have people studying this, the better information we're going to have and a better ability to actually tailor our pharmacotherapies for stroke patients. So challenge one, as I mentioned, your immune response, every single person in this room has a different immune response based on sex, age, modifiable and non-modifiable risk factors. So for instance, within the periphery, this is a great review, heart disease, hyperlipidemia, hypertension can actually induce chronic inflammatory statuses or immune cell composition changes. This can affect your gut. And we know from animal models, immune cells in the gut travel to the brain after stroke. So this is a systemic injury that happens. This stroke is not isolated to the brain. So when you have the immune cells trafficking into the CNS, they can actually exacerbate tissue injury, brain edema, lead to decreased functional recovery and higher mortality. So how is this altered? As I mentioned before, if you exercise, if you're young, if you have antibiotics or probiotic treatments, remote conditioning, so adaptive epigenetic responses that are changed, all of these modulate also the immune system and are not great in modeling the diversity of the human population in mouse models. Challenge 2, when we say B cells, it's just not B cells. That would be so simple and nice if it was just uniform, but there are subsets of every single type of immune cell that you have. So these are all the actions of a B cell. They can produce antibody, antigen presentation to activate your T cells. There's memory components. This is what mediates vaccines. And what we know after stroke is, for instance, the immunosuppressive aspect of B cells acutely is neuroprotective. You can suppress a lot of the innate immune response after stroke by producing IL-10 from B cells. But long-term, the antibodies actually contribute to cognitive decline. So now we have the same B cell population early on can be protective. And then if it sits in the brain long enough and converts to an antibody producing cell, can actually be detrimental. So how do we know when this happens? And how can we modulate this in the patients that we treat? My lab is also looking at the ability of B cells to produce BDNF and IL-10. So they can also produce neurotrophin production. This is the focus of my R01, but will not be what I'm going in today. But when we think about this, so now we've got an injury that's occurring, for instance, in our middle cerebral artery perfusion field. But what controls and supports recovery is contralesional homotopic neurons, subcortical neurons. You've got spinal cord plasticity after stroke. Throughout your CNS, you have areas that are undergoing plasticity. So what is our immune system doing in these other areas? Because typically, you think you've got an area of stroke, your immune cells are going to just cruise here and do their work. But that's not actually the case. And so one of the challenges that I have looked at in my animal models, which we'll go through in just a few slides, is where do immune cells diapedese in the post-stroke brain? And so here is a coronal section of a mouse, actually with serial 2-photon data, and this is co-registered to the Allen Brain Atlas, which is similar to co-registering to any MR atlas for human brains. But we wanted to really look at where B cells go. And so here you can see 2 little B cells. Ooh, that's right there in blue, that were pseudo-labeled in machine learning algorithm to pink. And this is in the contralesional hippocampus, which was strange and not what we expected, because I was pretty much expecting most of the cells to go to the infarct region. But in fact, they did not. And here you can see these are B cells, so we're talking about these are labeled B cells that were injected, and this is PBS, so all of the green here, is it showing up? Oh, good, it's showing up. All of the green is background fluorescence picked up by our automated quantification system, but clearly you can see that there are more B cells in this post-stroke brain. So right here is the area of infarct, and here is quantification of the pixels, so ipsilesional and contralesional. There are more B cells migrating actually into the contralesional cortex of mice that were B cell depleted, meaning that the other side of the brain is recruiting in B cells. So this is not what you would expect. And this can actually potentially be detrimental if they sit there for a long time. We just don't know that. What we were able to do, though, to orient you here, this is the mouse brain, and here's our area of infarct. When you co-register this with the Allen Brain Atlas, you can see that B cells, for instance, moved bilaterally. Here's the animal data into the dentate gyrus, which actually, when we depleted the mice, attenuated neurogenesis and induced memory deficits. So there's a protective role for B cells in this bilateral recruitment into the hippocampus. And they also recruited into motor function areas that induce motor function. So I won't go through all of this, but you can see a really nice bilateral movement of B cells. And you would not actually get this in standard histology. So I kind of skimmed over the methods, but this is taking a whole brain and sectioning and going through the whole brain to look at where every B cell has gone into in this mouse brain. And we can see that there is a bilateral movement. So we wanted to determine if this is happening. This means that our immune response is not just localized to our area of stroke, our infarct and peri-infarct region, that this subset of adaptive immune cells are moving out globally. So we decided to redo the entire experiment, this time with labeled CD8 T cells. So remember, these are the B lymphocytes, and your CD8 T cells are the ones that will target a cell that is at risk of dying or needs to die if it's infected, if it's tumorigenic. And when we did that, just to orient you down here, here is what the B cells look like, the pattern of diapedesis after stroke. And here is what the cytotoxic T cells look like. So it's a very different pattern. And when you look at somatosensory cortex after a stroke and after primary motor cortex, you begin to see that the CD8 T cells, they actually move into the injured hemisphere. So we are now parsing out different aspects of the immune system and where they will go after stroke, because this is critical. This area, actually, your somatosensory and primary motor cortex, your brainstem that's mediating sensory and motor related recovery, these are now getting detrimental CD8 T cells that if we deplete these cells, we actually have improved recovery. This is what I'm talking about when thinking about the precision medicine aspect of treating stroke. Where does the stroke occur? Which cell types are activated, and where will they go? Because in some cases, you may want to boost protective B cells if they're moving out into areas that support memory and cognition. But in other cases, you may want to actually deplete a B cell or a T cell population that may be impeding recovery and functional plasticity. And I say this because there is no way my lab will ever be able to do all of this work, but there are so many FDA-approved therapeutics for multiple sclerosis, ALS, long-term rheumatoid arthritis that target some of these populations that could actually be used on an individual basis if you can couple it with rehabilitation for a patient. So to summarize the first part of this, we have long-term plasticity that supports recovery in the post-stroke brain. It can occur in the infarct area, but really it's the rest of the brain that is supporting recovery. And we don't know how long adaptive immune mechanisms last. But we do know that they can be modified by risk factors and lifestyle. So both modifiable and non-modifiable. And in mouse studies, we see that B cells move into remote regions supporting motor and cognitive recovery. But the patterns of diapedesis are unique to the responding subset. So this is all of my mouse data that I'm going to show today, because the rest of it's going to be clinical. But I wanted to just sort of set in motion what I'm trying to do from an animal point of view is identify where and when responding subsets are going into the brain that actually support recovery. So with that, I'm going to go through two more data sets that are clinical. And this is going with challenge 4. So we've got modifiable risk factors. We have our B and T cell subsets are where they are going and which subset is responding. But this is absolutely a challenge, particularly for somebody who is not a clinician, because I need to be able to work with clinicians to understand and assess B and T cell subsets in patients. Fortunately, and one of the main reasons that I actually moved to Kentucky is because of the robust clinical research program that they have there. I mean, it's really been very foundational for me. And so this, as I mentioned before, is work with Dr. Justin Frazier and Dr. Keith Pennypacker, who originated Backtrack. I would love to help if anybody is interested in this program and what we do. But basically, this is during large vessel occlusions. So during mechanical thrombectomy, you come in, you put in the retriever, and as a general practice, blood is drawn back once the stent retriever goes past the clot in the brain. and pulls back intracranial blood right here to make sure that they've passed through the clot. And generally, this is discarded. So what Dr. Frazier and Dr. Pennypacker thought to do was to actually collect the blood in the OR. And we also collected carotid artery blood as control, and then there is the clots. So with this is a prospective enrolled trial. I believe we have about 150 enrolled patients right now in there, but this is including intracranial. Then you have systemic blood, which is how this will be shown in these data. And we also have banked clots as well. So with this, we have the company, we have 5 provisional patents right now on identifying clusters of biomarkers that can predict, for instance, cognitive decline in our patients or malignant edema. So I am not going to be showing most of those data. I will actually be showing what we did in our lab, which was develop protocols to use flow cytometry in the intracranial blood, which is usually under 1 ml of blood that is collected. So this is very small volume and took a bit of time to actually develop. But from our patient data, we had, for this first paper that came out, we had 16 large vessel occlusion subjects with matching intracranial and systemic blood samples. And we only use first pass blood. So if there is any complication and they have to go back in, we will not actually include that. And we compared this to healthy controls, so 12 healthy controls, as well as patients with cerebrovascular disease undergoing angiography in our clinic, generally for carotid artery stenosis. So this is the patient demographics for our stroke patients. We had 56% were female, decent range of BMI there. Most everybody had hypertension or some form of cardiovascular disease. And here is the NIH stroke scale on admissions. So we had moderate or moderate to severe in this population that I'll be showing you, as well as good perfusion success, full perfusion in 63% of the patients. So I will also talk about last known normal in a couple slides coming up. But we have these samples and we have been wanting to run flow cytometry in them. Absolutely happy to share any and all protocols that you see here, and as well as any protocols for analysis. So when I say flow cytometry, and I'm talking about this is actually taking immune cells and labeling them with fluorescent antibodies. I apologize if you all know what flow cytometry is, but I just wanted to make sure that we were on the same page. And you run it through a flow cytometer with a laser light source that will measure the scatter and the fluorescence of each of your antibodies to get this. And this is how data actually appear coming off of a flow cytometer. If you've ever seen these graphs before in presentation, each dot is a cell. And you can actually then look at the live cells, and we use a live cell marker. So you can tell which ones are alive and which ones are dead. And then we use CD45 to look at all of those cells that I showed you before. So your innate population, monocytes, macrophages, neutrophils, as well as your B and T cells of the adaptive immune response. And we use TISNE, so an unbiased algorithm, to actually use a principal component analysis to identify the samples. So this is the meat and potatoes of what we found. So I'm going to walk you through it so this is all 40 subjects, a compilation of the immune response in all 40 subjects. We had a flow cytometry panel that analyzed all of these cell types, and these are the ones that came up. They are color-coded here based on the population. So you can see, for instance, this salmon color is CD4 positive T cells, your T helper cells. Your B cells are this strange, I'm a fine arts major and I can't even think of what color that is, like a seagrass sort of color. And then you have NK cells in different populations. So all of your data is shown right here. Now, our 12 healthy patients, this is what their immune profiles look like. And this is how you can use TISNE analysis to actually identify your different subjects. What we have is a very different set of activation patterns in patients with cardiovascular disease. So remember, these are mostly carotid artery stenosis patients. And you can look, there's a specific CD4 T cell population. If you match it up right here, there's some CD8 T cells that are super high. So red is a higher population. We also have a population of macrophages in these subjects. So these are our baseline control samples in C and in D. Now we have a stroke onset, and we have this blood coming out in the ER or the OR with the mechanical thrombectomy. And what was pretty amazing was for the blood that is sitting intracranial, so this small patient blood volume right here at the end of the stent retriever looks almost exactly like your cardiovascular disease control. This makes sense though, right? Your blood is stagnant. It's sitting in there. This is what's happening. This is where these patients had cardiovascular disease ahead of time, and then the blood sits in the brain, and you have a general population activation almost everywhere, but the major clusters are still the same as those in our cardiovascular disease patients. But what's happening in the rest of the body, so this is within patients right here, you have a massive upregulation of everything else, similar more to a healthy control. So what do I do with these data? I have some very specific data to show you, but this is a lot to unpack, I suppose would be the way. And we publish these data, but what we need to understand is follow-up potentially, and I'll show you a little bit of longitudinal follow-up, but we just need more patients, I think. You know, with 16 patients, we can't exactly test for sex-based differences or do age-based differences or really parse out those who have type 2 diabetes versus those who do not. So this is where I love to collaborate and move forward on this because patient volume-wise, we actually need more to be enrolled. So when we go through, I'm showing you some of the innate immune responses. So remember, here's your innate immune response that occurs hours after onset. So this is exactly the time point that we have collected the blood from these stroke patients. And here to give you, this is our cerebrovascular disease control patients in green. Our healthy control average numbers are a dotted line. And then the within patient are these two bars right here that are shaded gray. So red is the systemic response, and then blue is the intracranial response. And here is the percent of whatever type of cell this is within the blood. So here you have your monocytes, your macrophages, and your granulocytes. So monocytes, macrophages, granulocytes, or neutrophils are shown here. And what happens is there is a massive drop in some of your innate cells. Now, what does this mean? If you look at the animal data will show that the first cells moving out into the brain after stroke are your innate cells. Remember, they don't need an antigen there to respond. They respond in general to everything. So we might actually be catching in real time innate cells moving out of the blood and into the brain of stroke patients. So we have this, and we specifically looked at some of the actual biomarkers in these patients. So remember, we can't parse this population out by sex or by age. But if you look at the general population in your systemic blood and the intracranial blood, the level of CD4 T cells that were there at the time of collection actually significantly or almost significantly related to malignant edema following 2 days after. So we have a potential pathological population here that is elevated in some of these patients. We don't obviously have a pre-stroke within patient control for this, but we have edema responses. Your NK T cells, so this is a different T cell population, was elevated based on BMI, and this is the systemic response. So now we're starting to get into our comorbidities and that these populations are elevated with elevated BMI. And finally, we have some of the dendritic cells. Classical dendritic cells shown here are depleted or lower as you have, are higher with time from last known normal. So now we get into last known normal aspects, particularly for our patient population in Kentucky, is huge because we have about a 200-mile watershed region for from spoke-in hospitals for our stroke patients. So It takes sometimes 3 or 4 hours for a stroke patient to actually make it to the university, which is one of only 2 comprehensive stroke centers in the state. So we take all of Appalachia, all of mountain driving through tiny roads to try to get it in here. So this is potentially a population that if we had more data and could understand this better, we might be able to try to inhibit. So with this, we also, in these data, and please stop me if there's any questions or anything. With this data, we also quantified endothelial cells and platelets. So one of the main concerns with collecting the blood in the intracranial portion with the stent retriever is that we are collecting, we could be damaging the vessels. So we put in a different flow cytometry panel endothelial cells markers to make sure that we are not injuring the patient vessel when we are removing the blood. And so here you see just a general flow plot. Along this line is your CD31. So here's your endothelial cells. There are virtually no endothelial cells in these. And then this is your CD45. These are your leukocyte populations that we just showed. So in the systemic blood that we've collected from the carotid, no endothelial cells. In the intracranial blood, however, we have one patient right here that clearly shows a pattern of increased endothelial cells within the blood. This was 1 out of 16 patients that they are shown right here, so there was no significance. This one actually more looked like artifact on the flow cytometry data. But the question, so what made this patient the only one with endothelial cells in the intracranial blood? And this goes back to biomarkers, right? So we're looking for biomarkers, trying to understand biomarkers with injury. And this patient actually on day 4 post had a massive hemorrhagic transformation. So this was malignant. Unfortunately, the patient died 2 days later. But this is the only patient that had endothelial cells within her intracranial blood, suggesting there may be a pathology in the endothelial vasculature to begin with that makes it fragile, right? So you now have a potential biomarker that we could use in the OR to look at any patient that is possibly at risk for hemorrhagic transformation. I am not a clinician, so I do not know what you would do with that if you could identify that patient, but this was ours. So to summarize how we're looking at these immune biomarkers during large vessel occlusion in this backtrack study, we have confirmed that endothelial cells are absent from the majority of the intracranial samples. We have myeloid macrophage populations that decrease in the intracranial blood. And as I mentioned, this tracks perfectly with the animal data. This was the first time anybody's ever shown this, that you could actually be losing populations that are moving out into the brain. You have T cell representations are increased and reflect pathology. So they reflect, or they can identify for higher populations, those who would go on and have higher edema. And I did not show you, but decreased neutrophils coincided with more neutrophils in the clot. So this was one question that we had from the reviewers. If your neutrophils are dropping, are they going into the clot or are they going into the brain? And we did show that there is more in the clot. So what we're trying to do now is identify specific adaptive immune cell subsets. So going back to our B cells, regulatory B cells are good early on. The plasmablasts, the one that produce antibodies, are bad long-term for cognitive decline. So we now have a new flow cytometry panel that we are doing in a next set of backtrack patients that are looking specifically at B and T cell subsets. So as I mentioned, and what I think is the other major challenge in trying to do any of this in a patient population is longitudinal sampling. And I don't know how it is here, but at our university, when they leave the acute care and go to a rehab hospital, this is not the same IRB, this is not the same university, you cannot track them. And unless you have an IRB specifically with somebody who's in PM&R and able to follow these patients up, we have a very hard time. It's a challenge to try to get the patients back. So we instead actually looked at some longitudinal identification of immune cell subsets in patients with aneurysmal subarachnoid hemorrhage. So this is in collaboration with some researchers I'll show on the next page, but these are all patients that had an external ventricular drain. We collected CSF and later on also processed blood, but this is actually one sample of CSF that we did receive. We again tagged everything for flow cytometry and looked at the populations. So this was CSF collected from the EVD at days 3, 5, 7, and 10 and analyzed by flow cytometry. I will say one of the challenges I don't actually write on here is for anybody who has, anybody done flow cytometry samples here before? Yes. We have to process all of this fresh, really. So you are talking 6 to 8 hours after whenever you get the patient sample. So this is a labor of love. I don't know if my grad students would call it a labor of love or one of the levels of hell, but this is also something that is a bit of a challenge because these data are absolutely best if you process immediately. So with this is actually in collection or in a collaboration with Dr. Kevin Hatton, who is a neurocritical care doc in the anesthesia department. And we were able to actually finish this up because Dr. Barb Nikolajic and Dr. Adam Backstetter had been working with Kevin on a separate process with these same patients and collected the peripheral blood. So this was pretty exciting because we were actually able to match our CSF and peripheral blood samples. This is for the patient-matched ones I'm going to show you initially. This is only 6 subjects. It took us almost 3 years to get 10 patients enrolled in this. Part of that was COVID, and COVID shut down, but part of it is just the volume of patients that we have here. So these are the final data. This is all unpublished now, but we were going to be hopefully getting this out in December. So you can see from our patient samples, we only had 1 male and the rest are female, a variety of BMI, but this goes back to small volumes. You can't really look, obviously, at sex or BMI. Some of them did have vasospasm. Oh, actually, all but 1. And a delayed cerebral infarction, they had 2 that had that. So we don't have enough in any cohort to actually look at some of the specific secondary comorbidities with the subarachnoid hemorrhage. But we did look at our immune populations. So here again is one of these plots to show you, which is the unbiased clustering of our flow cytometry data. This is all the CSF populations, and then the peripheral blood populations within the same patient. So you can see this is neutrophils actually, and that's more because we processed these immediately after receiving them from the hospital, and these were banked and killed the majority of the neutrophils. So that's an erroneous change in population. But even some of these other ones, you can see clearly that there is a lot more in the peripheral blood than these specific populations that are not present here, that are in the CSF. So this is what we're trying to do, especially also with the rapid postmortem brain, is trying to identify which patient or which immune cell subsets are in these samples. So I'll first show you some of the monocyte and macrophages. Remember, these are the ones that were also changed early on in the intracranial blood. So black here is shown in this population right here. And then this is looking at post-bleed days. We did not have a day 14 for the CSF samples. And each patient is actually color-coded between their CSF and their peripheral blood to help you follow along if you want to look specifically. But we did have a significant increase in post-bleed day within the CSF of monocytes and macrophages. Now, this is you have to be very careful with what you say. Is this new innate cells being recruited in? Are they leaving the brain and going into the CSF to be removed via the lymphatic system? What's going on? We do not know, but we do know that this is significantly increased and really almost unchanged within the periphery at the same time. So we have this for our NKT cells, which were also important with the intracranial. That is this population right here, color-coded. Now here is a very interesting, much higher representation within the CSF versus the peripheral blood. So now we start going into something that may actually be important for pathology moving on, especially because it significantly increases over time. And this though, again, if you want to get even more into the weeds, you now have skull bone marrow that produces B cells and T cells. And there's a question as to whether the majority of the immune populations after brain injury are actually coming from our skull bone marrow and not our periphery, which means then that any peripheral drug, which I talked about would be awesome to do, might not actually deplete the immune cells that are going directly into the brain, because there are skull bone marrow channels through the dura into the brain that may bypass any movement into the circulation. So we've got the NKT cells and then your B cells, actually, which is what we are very, obviously focused on. That is a significant decrease over time within the CSF. And this is your population right down here in red. So we have differences within patients. I would say the NKT cells are the ones that are seemingly the most critical. We had then the full population for the peripheral blood, and so we are actually now, I'm not going to show you all of these data, but this is the full cohort of patients that we had in our clinic over, as I mentioned, almost 3 years for trying to recruit this project. And what we are looking at are the immune cell subsets. So here you can see some of the data. Remember, this is the adaptive immune cells. So here you've got a cluster of B cells. CD19, CD11C B cells is something we're looking at in our animal model, because these are age-associated B cells. And they're there, but they won't respond in the same way to an antigen. So they're an interesting population that may be pathological. And we have other than populations within the CD8 and the CD4 T cells. So with the adaptive immune mechanisms, we're trying to move forward. We're trying to identify what is important. So I will summarize this with, we have higher levels of B cells in the circulation versus the CSF. And for the patients in all but one, there's decreased over time. So what does this mean? Maybe they're not as functionally relevant to subarachnoid hemorrhage over the course of time, at least from the CSF point of view. But our NKT cells was the only population significantly higher in the CSF, and that was maintained all the time that they were on their EVDs. So this may be a relevant pathological population. Unfortunately, as an aside, we did not have any IRB to actually look at the long-term recovery of these patients. So that would be nice to do. And now we are going into this subsets. So just to wrap up with this next three slides, why does this matter, right? So I'm trying to understand the immune cell populations that are responding in humans, sort of matching what I'm looking at in animal models of stroke. But we're beginning to really understand what is happening intracranially with our backtrack. So this is a summary of a lot of Dr. Pennypacker's data from our backtracks patients, but we can see migration is activated in the intracranial blood. Some of the cytokines contribute to angioedema. IL-9 can contribute to infarct size. So this is critical, because if we can really get a snapshot of what's happening, you could actually give a small drug while you are doing mechanical thrombectomy. You could give an adjuvant at the same time you are removing the clot and potentially neutralize some of the activated cells that are sitting there in the area or can be treated and have moved out. So with this, we are looking at this. But as I mentioned, our population is, a lot of it is Appalachian. So we've also been using these biomarkers, and we cannot do that with the flow cytometry that I have yet, but Dr. Pennypacker has a lot more patient volume. So we can look at the protein expression differences between the patient groups and their systemic response to stroke. And I'm going to just go through this very briefly set of proteins right here. So we've got, let's go back to our intracranial response right here. When you take it out of the brain, it is identical between these two groups. So remember, this is very stagnant blood. It looks similar to the blood profile of somebody who has cerebrovascular disease. So this is all well and good. But when you actually look at the systemic responses, just based on your zip code of where you live, matched comorbidities, matched age, matched severity of stroke onset, you have a very different systemic response. And this systemic response is similar to what would be against an environmental toxin. And I'm not going to go, into details, but this is a chemical response in the blood of our patients that's very different just based on their, unfortunately, their zip code. So now we get into why does this matter and what complexity does this even add? If you are in an area that has poor water sources, you are in a food desert, what does this do to your inflammatory response and how does it complicate your recovery after stroke? So, going back to this, I started this whole thing with the 2006 article that came out that said, you know, there was no hope. They had over 1,000 compounds in animals that were successful, tested over 100 in stroke patients, and nothing worked. I'm still hoping that something will work eventually. And this is why I think that identifying immune-based responses to stroke and how they can be altered in the individual patient by comorbidities, by where you live, lifestyle factors, and that can really move us forward on looking at rehabilitation and immunotherapies that can support rehabilitation. Ultimately, that's a much longer time window. So a few things to consider. If this is all you pull from this lecture, the immune response is affected by everything. You have 800,000 patients. I don't know how many patients a year you have here, but the immune response is affecting the way they are injured and the way they recover. It is unequivocal. And it also, all of these comorbidities, everything affects which immune cell subset is actually activated by the stroke and which one responds to it. We just need to understand how to identify the bad ones. And we need a better understanding of long-term adaptive immune responses. Vaccines work. There's a reason vaccines work, because we have memory in our immune system. That means that the first stroke induces a memory in our immune system that will affect how we respond to second, third, and fourth strokes. And we also need to identify the responses linked to injury, cognitive decline, and functional recovery. Much more difficult to do in patient populations because of the required number of enrollees. But as I mentioned, I absolutely think the longer time window may help in therapeutic development. I think that adjuvants to rehabilitation are going to eventually be what is necessary for an individual to improve recovery. And with that, do you have any questions? Thank you.
Start. Great. Thank you, Ann. So many thoughts that have to come from this. We've just heard that you need to be thinking about personalized medicine. Every patient is different, and they're different really in many ways. Their genomics, their proteomics, but the immune system, which adapts constantly, this is how you are continually becoming different. This is going to be a key to health. Immunotherapy, immunology as a biomarker before its therapy, that's a key. Let me ask a question about a cell which fascinates me, which are microglia. Are they just misnamed and you've been studying them all along? Or when we look at the tissue, will that be a profound addition to this?
Yes, I think that's a great question. And microglia are so complicated. I don't, a number of circulating monocytes can actually go into a microglial phenotype once they move into the brain. So potentially in the backtrack, you see the monocytes that are moving out into the blood. That's a great question, actually, because you could almost isolate those and try to drive a microglial phenotype and see what they're doing. But I think that a lot of what we see in the periphery and the circulation is not microglia, but potentially monocytes that could actually. And microglia can, they can, you know, phagocytose synapses and clear out during plasticity as well. So yes. Yeah.
Questions for Dr. Stowe?
Yes.
Very nice talk and really like wondering your biomarker studies have done very nice in your protein biomarkers. Identify any specific
Yes, and there are actually a number of them. I'm happy to send you some of the references. So this is Dr. Pennypacker's work, but we do have IGF, I can't remember, there's a panel of 5 actually that we see with machines. So this has been done with machine learning algorithms. So for anybody who's interested in patenting biomarker profiles, you can't actually patent the profiles, you can patent the machine learning algorithm that identified the profiles. So this is where we've gone, and there are a number of different profiles for different aspects of edema, and then infarct volume. Cognitive decline is what we are also looking at there. So I don't know the specific protein profiles, but I'm happy to send them to you if you'd like. You're welcome. Yes.
In one of your slides, you showed that normal brain, there are not that many olfactory bulbs. After stroke, there are lots of B cells in the olfactory bulb.
Yes.
I'm just wondering, what is the functional significance? Is it related to neurodegeneration?
Anosmia, is that the without smell? Actually, I think there's some stroke patients that can get anosmia. That's interesting. So I don't know what the functional aspects of that, but there is an induction of proliferation and neuronal progenitor cell migration through that track in mice because it's very important. So some of that may be a mouse-specific because they are driven predominantly by smell. And so if they have an anosmia, they might have an inflammatory response there. But then you get into lymphatics also might play a role there from that. But I don't know why, actually.
There's a lot of interest in the support cells of the olfactory tract as opposed to the neurons themselves as being the immunologic key to the loss of smell with these disorders. So you're exactly on target.
Yes.
Yeah.
Then also a bit confused how we correlate between humans and rodents, because one of your slides you showed that body mass index cell population, but in rodents we have seen similar phenomena, nothing to do with body mass index.
I know, and that's why I always try to do everything as much as I can in clinical patient samples, because you're absolutely right. With a mouse, you know, we're generally using black 6 mice, which are supposed to be genetically identical. And if you follow the 6J, that's only 6 generations from the founder line at Jax. So there is no genetic diversity at all in what we do in mice. There's no BMI differences. We are looking at aged animals now, which is a challenge with inducing a stroke in an aged animal. I still do all of my own stroke surgeries in my lab, but yes, that goes back to what I would love to be able to do a gadolinium or some sort of blood-brain barrier disruption in acute with the inflammatory biomarkers to see if you could look at lesion areas within the brain after stroke or potentially blood-brain barrier disruption, kind of to get to how do we study that in humans, because it's not going to happen in mice. But
Dan, you have a question?
yes.
Two thoughts, a little bit of timeline. It's good to be but it's too long. These cells you're mentioning, take the long term or whatever.
That's great. So I'm going to base this on mouse studies because that's what it is. I would say acute 4 to 7 days. So now you get into the activation of the immune response. And CNS antigens are everywhere. So generally, our immune system is barred from actually seeing neuronal antigens and myelin antigens. That only happens when you have an autoimmune disease like multiple sclerosis, where you are attacking your myelin. But if you were to survey half the people in this room, we would actually have myelin activated immune cells without any autoimmune disease. So what I think is happening is the good cells come in, they can produce neurotrophins and support and be anti-inflammatory and support recovery. But if they stick around too long and then they meet antigens and have germinal centers form, which we know occurs in the brain and in the dura, then those cells could actually be producing antibodies to neurons, for instance, if it's long-term over time. And those antibodies then could induce opsonization or other cell death mediated pathways within the brain. And I just proved my second R01 hypothesis wrong. They're never going to fund me really by the time this is all done, but because we depleted B cells in aged mice, so these are 18 months or older, so 60 to 70 to 90, we had some old mice. And we thought we did a Pavlovian auto shaping task. So the mice used touch screens to do Pavlovian auto testing. And we depleted the B cells thinking, this is going to work. And so originally, like at the 2-week time point, those who had B cell depletion were actually worse. But then at the 4-week and 6-week, you watch them get better. And the ones that had B cells got worse. So there you go, right there. That is the transition. So if I had to guess, I would guess your 4-week time is the transition where things go bad.
Students, don't ever be afraid to prove your hypothesis wrong. That's how you learn. And when you write the grant, you say, if this is true, we're going this way. If this is proven false, we're going this other way. That'll be important.
Yes.
And thanks very much for getting us started, getting us thinking about huge topics. This continues all day and the rest of your careers. But Ann will be here today and helping us look at the posters, etc. I have a few gifts for you. First of all, Dr. Gilboe has been very important to us, and this is a special talk. So this is a plaque of your lecture with our great thanks for being here.
Thank
That's
you.
very
I've
good.
forgotten the
And
plaque before.
that's a
Thank
picture.
you.
Well, it's high time.
So cool. There we go. This is going up in my office.
We have some more tangible things as well that don't have to go up in the office. Apparently, everyone is getting a little certificate, so
Awesome.
thank you.
Thank you.
There's a little remembrance of Wisconsin and a little more tangible because Dr. Gilboe's lecture has a monetary award as
Oh,
well. So
thank you.
thank you very much.
Okay, thank you so
It's
much.
our pleasure.
All right.